Good morning and welcome to, thank you, and welcome to this Friday morning session on environment, nutrition and cognitive development. And maybe I will give a brief introduction about metabolism, brain energy metabolism, and how that relates to the presentation of the morning. So, as you probably know, and in fact, the chairman of Nestle mentioned it last night in his speech, the brain, despite the fact that it's only 2% of the body weight, uses essentially 20-25% of the energy, whatever index you take. So it's a relatively high energy demanding organ. Now, in addition to this feature, which defines brain energy metabolism at the global level, there is another important feature, and that's the regulation in space and time in register with activity. And in fact, the first formulation of this was by Sherrington himself at the end of the 19th century, when he postulated that the brain possesses an intrinsic mechanism by which its vascular supply can be varied locally in correspondence with local variations of functional activity. And we know now, with the work of Lou Sokolov in particular, that actually this is true also for glucose utilization. You see ocular dominance columns, the primary visual cortex, that are defined by the increased glucose utilization. Now, in summary, high metabolic cost and highly regulated in space and time in register with synaptic activity. And by the way, this has been at the basis of the developments of the techniques for functional brain imaging, because either PET, for which one can monitor glucose utilization, oxygen consumption, or blood flow using oxygen-15 water, one monitors metabolic events or vascular events. And even fMRI, in fact, is related to vascular for sure, but also metabolic events. And it's actually the change in oxydeoxyhemoglobin that provides the intrinsic signal. So all these techniques that are widely used, we have seen examples yesterday, and we will certainly see today again, all measure indices of metabolism, which hopefully are related to the electrochemical events that occur at the synapse. And actually, this has been a key issue to try to understand, and in particular, my group has been interested in this, is trying to understand how these electrochemical events are translated in a metabolic and vascular response. And as I mentioned a moment ago, also this provides a better understanding of the origin of the signals for functional brain imaging. Now, the classical view until the early 90s or mid-80s was that, in fact, neuronal activity would release some sort of vasodilator that could increase locally blood flow, and that would deliver energy substrates to the active neuropil. But in fact, this was not taking into account an important player in this cupping. And these are astrocytes, a type of glial cells. These are drawings from Cajal himself. These are more modern views of astrocytes. And Cajal, in fact, has already identified the fact that these astrocytes also possess processes that are in contact with the blood flow and with capillaries. And in fact, Golgi, even before, was proposing that this could mean that these cells could play a role in, as he said, distributing nutrients to the parenchyma of the brain. Now, these astrocytes have also interesting features. Here you see a 3D reconstruction from EM stacks showing a postsynaptic profile, a presynaptic profile, and here in blue, this astrocytic lamellae. And in fact, synapses are, not all of them, and it's also dynamic, this coverage can change, but essentially they are ensheathed by these astrocytic profiles. And on the other side of the astrocyte, if I may say, you see here in negative, actually, a capillary stained because of the astrocytic processes called ENFIT are actually defining their structure. So in a more artistic view, here you have the relationship of these cell types. The neuron the capillary, the source of energy, the major consumer of energy, about 80–85% of the energy is consumed by neurons. And here, around the synapses, these astrocytic processes, which, by the way, express receptors and reuptake sites for transmitters, so they can sense synaptic activity. And here, on the end feet, they express transporters for a number of substrates. So one can appreciate that these cells most likely play some kind of role in coupling synaptic activity to energy delivery to the parenchyma. So a few years ago, almost now 2 decades ago, we described a mechanism by which this coupling can occur, being mediated by glutamate. In fact, we showed that glutamate can promote glucose uptake, and that was the big surprise, which for a long time was difficult to convince people, but I think at the end, because of our work and work of others, it became clear that this is an important molecule. Actually, they take up glucose and they release lactate. So they are actually the site of what is known aerobic glycolysis, a well-known process known in cancer cells as the Warburg effect, meaning the production of lactate in the presence of oxygen. So this mechanism provides a pretty straightforward mechanism to couple synaptic activity mediated by glutamate, and we're talking about 80% of the synapses in the cortex, for example. And this glutamate uptake, which uses the electrochemical gradient of sodium to drive glutamate and remove it from the synapse, that uses energy, because sodium is extruded, because glutamate is recycled. I will not go into details. And this provides then a signal to import glucose and activate this aerobic glycolysis. Now, this process now is known as the astrocyte neuron lactate shuttle, and we characterize several of the players from the glutamate transporters, the alpha-2 subunit of sodium potassium ATPase, glucose transporters, the different LDH lactate dehydrogenase form, the monocarboxylate transporters. The end message here is that, first of all, there is a neuronal signal that triggers entry of glucose into the parenchyma, and there is a transient aerobic glycolysis in astrocytes because lactate can then be transformed to pyruvate and then provide energy because there is oxygen. But essentially also it provides, and there are several evidences for this, that astrocytes are mainly glycolytic, so they produce lactate, and neurons are mainly oxidative. Now, there is another interesting feature about astrocytes in relation to brain energy metabolism. They are the only cells that express glycogen. You see here a capillary, you see here an astrocytic end foot, and you see it is filled with the electron-dense granules, which are glycogen, and you see here the neuropil. So in summary, what we know now is that there is definitely a key role in brain energy metabolism by these cells, astrocytes, that they essentially produce lactate either through glycolysis or through glycogenolysis, both leading to lactate production. Now, more recently, in particular, in collaboration with Christina, and Christina Alberini, who was the first speaker this morning, it turns out that lactate is not only an energy substrate, but it is also a signal. 3-4 years ago, we published this paper showing that astrocyte neuron lactate transport is required for long-term memory formation. And Christina will elaborate on this and other aspects of her work. But we followed up on the molecular side and found out that actually lactate is, as I said, not only a signal, but actually it's a positive modulator of the NMDA receptor, and this actually results in the induction of plasticity genes. So you have a molecule, a primitive molecule possibly, lactate, that actually is energy metabolism and also a signal for plasticity. And as you can see, this is specific for lactate. None of the other energy substrates do this. And for example, lactate in neurons induces ARC, CFOS, ZIF268, and BDNF. And you can see here that it amplifies the NMDA current. It's just a quick example. More interestingly, following on this, we found by doing mRNA sequencing transcriptome analysis on neurons that actually lactate induces a large number of genes. So most likely through this NMDA receptor signaling amplification. And we're now trying to sort out what these genes do, and actually they seem to be related either to plasticity or to neuroprotection, which is quite interesting because there are examples that lactate is actually neuroprotective. Now, just a quick view of where these glycogen granules are in relation, because since they are related to plasticities, one might expect that they are present near synapses in astrocytes. So we did this work actually in what is called the CAVE, which is a 3D full immersion facility at KAUST, where by using 3D reconstructed stacks of a block of cortex, and then segmentation, identifying each cell type, astrocytes, blood vessels, neurons, axons. Then we were able to transform this into a 3D virtual reality. So it's really like you are actually taking a dip into the brain, and you see here the back of the capillaries. You start seeing here a synapse, and then in green astrocytes. And these purple dots here are actually glycogen. And they seem here, you see here a dendrite reconstructed. And they seem at random, but when one does statistics, one can actually see that most of these glycogen granules are present in astrocytic processes around the synapses. Now, the first speaker will be Christina, and she is from the Center for Neural Science, New York University, and she will present work, not done at the moment, with energy metabolism in long-term memory formation and enhancement. Now, to introduce also briefly the next speaker, I would like to go back to a point in this coupling between neuronal activity and energy metabolism and the existence of this aerobic glycolysis in astrocytes. And in fact, Mark Reichel, a few years ago, showed that during activation, for example, a visual stimulus, there is an increase in blood flow, an increase in glucose utilization, which is not immediately matched by a commensurate increase in oxygen consumption, meaning that paradoxically, there should be an increase in oxyhemoglobin in the activated area. And this, by the way, induced Seiji Ogawa to develop the BOLD technique, the blood oxygen level dependent imaging that is widely used now. But more recently, what Mark has shown, and it was mentioned by Mike Gazzaniga yesterday, is that this aerobic glycolysis is actually present also in certain brain areas, even at rest, under baseline. And when one makes a correlation between the regions where aerobic glycolysis is operational in the adult, which is essentially the default mode, this correlates with the expression of genes involved with synapse growth, dendrites, neuronal projection, axonal growth, everything that has to do with plasticity. So where there is aerobic glycolysis in the adult brain, there seems to be an association with the expression of genes related to plasticity and development. He calls them neotenous gene expression. And what is interesting is that actually in early development, the brain actually widely expresses this metabolic profile of aerobic glycolysis. And the postulation by, as Mike mentioned yesterday, by Goyal and Reichel, is that aerobic glycolysis, which is predominant in the brain, in the developing brain, may be related to synaptic plasticity and expression of genes related to this phenomena. And there is some coupling, some oxygen consumption, of course, but this seems to be more related to synaptic transmission. Now, in the adult, particularly in the cerebellum, where aerobic glycolysis is virtually absent, there is a full coupling, so mostly related to transmission. But these neotenous regions in the adult brain, such as, for example, a default mode, there is still some aerobic glycolysis and therefore an expression of genes that are involved in plasticity and development. Christopher Kuzawa will be the second speaker. He's from Northwestern University. And actually, he will deal with exactly at least one aspect of this, which is the energetic cost of building the human brain, implications for evolution of slow childhood growth, and the origins of diabetes. And finally, the topic of this morning is environment, and this is just an image from a series of very classic papers showing that the environment plays a key role in developing brain and in neurons. For example, in the density of dendritic spines, you see an enriched environment here, an animal reared in an enriched environment or in a poor environment. And as you know, these dendritic spines are highly dynamic. And so the role of the environment, obviously, and we again heard it in the nice presentation by Mike Gazzaniga yesterday, plays a key role in brain development, and actually we all know it. And Maureen Black from the University of Maryland School of Medicine will address this point, particularly from the angle of nutrition growth, and the developing brain. So, without further ado, after this brief introduction, it is my great pleasure to introduce Cristina Alberini, and she will talk about energy metabolism in long-term memory formation and enhancement.
Okay, great. Thank you, Pierre. I'd like to start by thanking the organizers for inviting me. It's a great pleasure to be here and an honor, and to have the opportunity to share with you some of the work ongoing in the lab, and particularly the project that I'm working on in collaboration with Pierre. So today I'm going to tell you about the studies related to energy metabolism in long-term memory formation and enhancement. And as you have heard already in the introduction, metabolism is very important for brain functions, not just to keep the cells alive, but is also an activity-dependent response. Brain metabolism affects how we feel, how we function, and how we age. And disorders of brain metabolisms are linked or may cause these devastating diseases such as neurodegeneration, like Alzheimer's disease, but also neuropsychiatric diseases, epilepsy, and even cancer. So having optimal brain metabolism is important for an optimal health. And despite all this knowledge that is mostly correlational and descriptive, it is actually interesting that not enough is known about how brain metabolisms are regulated. And Pierre has indicated some of those issues about energy metabolism. And the energy metabolism is one of the important in the brain because it supplies energy. And as we have heard now many times, the brain requires a lot of energy to function, much more than any other tissue. So we need really to understand how energy metabolism in the brain works, how it changes with different functions in different brain regions, perhaps, as we have heard now from Pierre, in different stages of life. In development is different. There's much more requirement of energy in a developing brain, and then in the normal function in adult brain stage, as well as in aging, and all the diseases that are related to that. So trying to understand the energy metabolism, we need to understand why all this energy is needed. And as Pierre has suggested, most of it goes to supply energy to neurons, neuronal activity, to keep neurons going. 89 billion neurons in a human brain, they require a lot of energy. They cannot do that autonomously, as we have heard. And so what is the answer? The answer is circulation. Blood is brought to the brain, and the vessels make sure that nutrients and oxygen reach every single cell in the brain. But again, neurons cannot just make the energy necessary for activity, especially during the cycle of when we are awake and we function, we think, and we operate. And therefore, they need the help of other cells, and astrocytes are very well positioned to do that. And you have seen this already. So astrocytes are positioned between blood vessels and neurons, and glucose is actually taken up by astrocytes and then delivered to neurons, actually in forms of lactate in an activity-dependent manner. So that is a very interesting mechanism for the brain to use as energy supply, and it's not just energy. As I'm going to show you, we see that if we supply equicaloric concentration of glucose versus lactate, we don't have the same effect. So definitely lactate is more efficient or supplies something in addition to glucose to the neurons to function. Why do we need all these interactions and collaboration among different cells to do that? As I mentioned, because neurons are busy firing and they cannot provide sufficiently the energy necessary for that. There is a lot of oxidation, and too much oxidation clearly leads to problems in the cells. Reactive oxygen species, oxidation, and if this is too much, is going to lead to cell death or cell damage. So the help of astrocytes is not only to supply the energy, but also to make sure to restore homeostasis. And lactate is helping in doing that. And in my lab, we have been studying the biological mechanism of long-term memory formation for about 20 years now, after training at Columbia with Derek Kandel on aplysia. And we're going to have the story in the evening. I look forward to that. We focused our questions on the mammalian brain because we wanted to understand how complex brain works in memories that are relatively modeling complex memory of human brain. So we have focused on understanding the gene cascade that is regulated in the brain and necessary for long-term memory formation, particularly in the hippocampus, a region that is important for memories that process facts, people, and things in humans, the so-called declarative memories, memories of places, context, space, and so on, and is a region that is very well studied. And as most of my other colleagues, we have focused in the last 20 years on the gene cascade that is regulated by learning and required for long-term memory formation, but thinking about neurons mostly. Because clearly we need to understand what is the gene expression that occurs in neurons to support long-term memory formation. But when we talk about the brain in vivo and a complex brain, and as we have seen, neurons are not the only cells that are working in an activity-dependent process, I became interested in understanding how this unit works together, and particularly astrocytes and neurons work together in long-term memory formation. I visited Pierre Magistrati's lab, I think it was 2008, and I heard all the details about his models that, as he already presented, proposes that astrocytes and neurons are coupled through lactate. Glucose taken up by astrocytes is going to be broken through glycolysis into lactate, and lactate transport into neurons is going to supply the energy and other things necessary for the neurons to process the information. I became interested in that question because it made very much sense to me that there is the need of the help of energy and astrocytes to do that. So we decided in collaboration to test the hypothesis. In addition, as Pierre mentioned, there is not just glycolysis coming from glucose, but astrocytes and not neurons in the adult brain store glycogen. and glycogen is the storage of energy. So when there is an activity-dependent process, glycogen is going to be broken down. It's going to be broken down into the glycolysis and supply lactate to neurons. So testing this hypothesis, we are working actually on a process known as consolidation. Consolidation is the process that makes sure that the learned information is going to become a long-term memory. Not all the information is important. We need to select the important information and to store the important information. And memory initially is in a labile state, is fragile. So several types of interferences can actually disrupt this stabilization process known as consolidation and prevent long-term memory from being stored, information from being stored. And this process is made of multiple phases. The initial phase of consolidation of long-term memories is a requirement for gene expression, de novo protein synthesis and de novo gene expression. And this continues for quite some time. And we have chosen to study the process of memory consolidation in the hippocampus using rodents. I'm going to describe the model in a minute. And the approach is, as I mentioned, rodents, mostly rats. The majority of the basic questions we're studying in the lab are done with rats. And for animal disease models, then we go to mice. We use a task that is simple, and it's a single learning trial event leading to a robust long-term memory. The reason why we have chosen that is because we want to identify what changes immediately after learning and follow up over time these changes. We need a single important event. And again, I'm going to describe the task in a second, and then we're going to follow up these events in brain regions. As I mentioned, particularly the hippocampus is one region that we have been interested in because our memory model models hippocampal dependent memories. The task is known as inhibitory avoidance or passive avoidance. It's a fear-based association. It's a fear-based memory task in which we have a shuttle box. One compartment is lit and the other compartment is dark. They're separated by a door. The animal is placed into the lit compartment. A few seconds later, the door opens, allowing the rat to enter into the dark side of the chamber. And once the rat is in that context, door closes and they get a food shock. And training is complete. It's a single learning event, a fearful event. They associate the context with the food shock by entering into the dark side. Memory is tested anytime, if we want to test a short-term or long-term times, by replacing the animal into the lit compartment, opening the door, and measuring the latency that they have developed. And you're going to see a lot of bars like this. Acquisition is the time that they take to enter into the dark chamber before getting the shock. So they only take a few seconds. They are in, they get the food shock. But after that, they're going to remember, if they have memory. And so when we test the memory, they're going to show a latency to enter into the shock compartment. And you're going to see that they develop a long and robust latency. This is a typical latency 2 days after training, mild food shock, 0.6 milliamp. And in this case, most of the rats don't even enter into the dark. The cutoff time here is 9 minutes, after which they are taken back to the home cage. So with this paradigm that reproduces contextual memories, but context associated with a strong emotion, fear, we are looking at regions in the brain and what are the biological changes occurring in these brain regions. We started, as I mentioned, from the hippocampus because the hippocampus is required for the acquisition and the consolidation of this memory. So we are taking out the dorsal hippocampi, which is the region that processes context shock association. We are looking at the changes occurring with learning. So we're not manipulating the brains. We're asking the brains of these animals to tell us what happens after learning, and we follow up these changes over time. And then we manipulate them by targeting the regions stereotactically, directly, with molecular manipulation, whatever is the information that we want to measure or we want to test. With molecular manipulation, we are looking, or pharmacological, we are looking at whether those changes are required for long-term memory formation. And with this approach, we have identified a number of players. The first hypothesis was actually to test that what we knew from invertebrates, whether what we knew from invertebrate system was also applying to mammalian brain and mammalian memory systems and consolidation that requires the hippocampus. And in fact, that was the case. The activation of the transcription factor CREB is required for the consolidation of this task. Another downstream transcription factor known as CBP, the induction of it in the dorsal hippocampus is required for this task. And we have then identified a number of target genes. So here I'm diverging a little bit from the metabolism, but I'm going to get back to it. I want to mention that some of the target genes that we have identified with these studies in the dorsal hippocampus are interesting nutrients. One in particular that I'm going to mention is insulin-like growth factor 2, which is downstream of CBP and is upregulated with learning. If we block this upregulation by knocking down the expression of IGF-2, insulin-like growth factor 2, we block long-term memory, so it's required for long-term memory formation. And if we supply IGF-2 with training, we actually boost the memories tremendously. Strong increase, enhancement of the memory, and long-lasting. We tested also the other factors that belonging to the same system. IGF-2, insulin-like growth factor 2, is structurally similar to insulin, doesn't have the same function. And the other member is IGF-1, insulin-like growth factor 1. And insulin, but not IGF-1, enhances memory if injected into brain regions. But the effect that we see with insulin is actually transient. So IGF-2 is, among the members, the one that has the strongest effect as memory enhancer and on also memory persistent. It also has a number of interesting effects in rescuing memory deficits. But I'm not going to go on with this. I just wanted to mention, because actually I learned a week ago that, maternal milk is rich with IGF-2. So that's an interesting... Okay, but let's go back to metabolism. And these mechanisms have to do, obviously, a lot with it. So we set out to determine whether the astrocytic neuronal lactate coupling is important for the changes occurring in the brain that I just mentioned that are needed for long-term memory formation, as well as long-term memory formation itself. And this question was addressed in the lab by a former postdoc, Akinobu Suzuki, now back in Japan with his own lab, and a graduate student, Sarah Stern, now at Rockefeller as a postdoc. So what they did, Akinobu and Sarah, they systematically asked the question of whether the glycogen metabolism and lactate formation and transport are required for long-term memory formation, for inhibitory avoidance long-term memory formation. And I'm going to just summarize because these data are published, and then I'm going to go to something that is non-published. So in the first set of experiments, they asked, if I block glycogenolysis, do I have an effect on memory? Do I have an effect on those changes that occur with learning? And the answer is yes, in both cases. So if they block glycogenolysis by injecting directly into the dorsal hippocampus bilaterally, DAB, which blocks the glycogen, breaking down the glycogen to glucose 6-phosphate, so this step. When they blocked that, they found that long-term memory formation was impaired, that this deficit was rescued by giving lactate, suggesting that lactate was the key molecule for this impairment. The molecular mechanisms, the induction of those changes that I mentioned, and we have looked at the numbers, phosphorylation, CBP, immediate early genes, and so on. Also, those molecular inductions that depend on learning were blocked by DAB, and again, rescued by lactate. So that was the first piece of evidence that glycogen metabolism is involved in long-term memory formation and the changes that underlie that. The second question that I addressed was to measure directly lactate intra-hippocampally with microdialysis. And what they found is that after learning, there is an induction of lactate, an increase of lactate in the extracellular space of the hippocampus with microdialysis that lasts for about an hour and goes back to baseline after that. And also that is dependent on glycogenolysis. So DAB blocks the increase of lactate in the hippocampus. And then they ask, if I block the transport of lactate by blocking specific transporters that are expressed by astrocytes and not neurons, and those are monocarboxylate transporter 1 and 4, and they did that by knocking down the expression of these transporters in the hippocampi of these animals. What happens? What they found is, again, memory loss. Long-term consolidation was impaired, rescuing with lactate, but not with equicaloric concentration of glucose, suggesting that lactate does something different than glucose. And then they went on and asked if we downregulate the expression, so if we knock down the expression of other transporters that are specifically expressed by neurons, those are monocarboxylate transporters 2, what happens to memory? And what happens is that memory is impaired. Now they supply lactate and they don't rescue the impairment because clearly the monocarboxylate transporter 2 transport lactate in the neurons. In fact, there was no impairment with that and no change with neither lactate or glucose. So all this data, in addition to LTP, long-term potentiation data, studies done in vivo with collaborators at Mount Sinai, George Huntley's lab, basically led to this first set of conclusions. And that is lactate is released with IA training in the hippocampus, blocking glycogenolysis blocks lactate release, long-term memory as well as the underlying molecular changes, and lactate rescues these impairments. Similar results are found with LTP in vivo. Transport of lactate from astrocytes to neurons is essential for long-term memory formation and for those changes required for long-term memory consolidation. Equicaloric concentration of glucose do not rescue these effects. So the conclusion is that glycogenolysis and astrocyte neuron lactate shuttling are required in the hippocampus for long-term memory formation. But why lactate? What does it do? So we need to get now to the next question. And the next question is, what type of mechanisms really require lactate? Well, we are thinking about activity-dependent mechanisms, but we really don't know. We need to find out which are requiring lactate and perhaps which may have enough of the energy supplies to carry on. So clearly memory we have seen, but a number of other functions that needs to be tested, brain regions and systems. And one that is a follow-up of our studies in inhibitory avoidance is the emotional modulation. Consolidation was discovered actually because memory, long-term memory formation can be modulated. So modulation strengthens the memory. And why do we have a consolidation of a single event? Is because that event is emotionally important. So arousal, motivation, stress, and emotional regulation are important, fundamental, to make sure that the representation of a single event is going to become a long-term memory. Otherwise, we will not remember. Do you remember what happened to you last week, day X, 4 o'clock in the afternoon? Unless there was something salient to that moment, we forget. And there is a vast literature that has studied the role of stress and arousal on memory and modulation. So if we have very low stress, we cannot form long-term memories, but we increase the level of modulation, we increase the level of memory strength, consolidation, and modulation of memory up to an optimal level, after which, if the stress is too much, then there is a negative effect on memory. So memory is going to be actually impaired. And this inverted U curve of stress on memory is a very interesting function, because we go from adaptive mechanisms to maladaptive mechanisms by changing the level of stress. So today we're going to talk about the adaptive part. Having an emotion, having a certain level of stress, and making long-term memory formation. And we know that this modulation, the stress modulation, requires the regulation of stress hormones, noradrenaline and glucocorticoids. And as I mentioned, a large body of literature have studied the inverted U curves or have documented modulation of memory. I'm mentioning Jim McGaugh, but many others are very interested in understanding this modulation. And the two stress hormones particularly involved in this modulation, as I mentioned again, is noradrenaline and glucocorticoids. We are interested in studying the role of both these hormones with metabolism, because clearly the brain is going to be very active. It's an important event. There is arousal. And noradrenaline is known to be coupled to glucose metabolism. So the hypothesis is whether noradrenaline is actually activating glycogen metabolism and decoupling to neuronal functioning. And the question has been addressed in the lab by Virginia Gao, a student, an MD PhD student, and she's asking, is noradrenaline in the hippocampus working through astrocytic mechanisms? So the first question she asked is whether if we block receptors for noradrenaline, adrenergic receptors, in the hippocampus, do we affect long-term memory? And this is, we predicted this outcome because a lot of studies have done similar experiments by targeting amygdala, which is another region that is very enriched of these receptors, the adrenergic receptors. And adrenergic receptors come in many groups. We have alpha receptors and beta receptors. And we are focusing on beta receptors because they have been described as important for long-term memory consolidation and modulation in amygdala and other regions. So now we are targeting the hippocampus. And she asked, if I block beta adrenergic receptors by injecting propranolol, an antagonist of beta adrenergic receptors, before training, what do I see? And in fact, what she sees is that memory is impaired. We test a day later, so long-term memory. We test again 6 days later, a week after training, to make sure or to test whether the effect is persistent. And the effect is persistent. So memory with propranolol on board is impaired. The effect is persistent. If we inject lactate with propranolol, memory is rescued. The effect is persistent. If we inject equicaloric concentration of glucose, the impairment remains. So again, like with DAB, memory is impaired if we block beta adrenergic receptor in the hippocampus, and this is rescued by lactate, but not glucose. The next question was to look at the molecular mechanism required for long-term memory formation. I'm going to show you only one. We look at several, and I'm going to show you one which is neuronal. It's the induction of the immediate early gene ARC. We know it occurs in neurons and in excitatory neurons in particular. And as you see here, the expression of ARC is induced after training, is significantly increased in the hippocampus of these animals. Injection of lactate alone does not change this induction. Injection of propranolol completely blocks the induction. Injection of lactate with propranolol rescue the deficit. And this is true for many other markers. She went on and then asked, are beta adrenergic receptors important for the release of lactate evoked by training? So she measured with microdialysis in the dorsal hippocampi, and she actually wants to test whether beta 1 or beta 2 type of receptors are involved, perhaps both, are involved in this regulation. So in this case, she measured, these are naive rats injected with vehicles. She measured the level of lactate over time, right? So there is a measuring before training, and then she continues after training for 90 minutes. and this is the baseline. Training significantly increased the level of lactate in vivo for about, as I said, 60, 70 minutes, goes back to baseline after that. This induction of lactate is completely blocked by propranolol in the green line, so lactate levels remains flat with propranolol on board. And then she tested betaxolol, which are selective antagonists of beta-1 receptors, and found that there was no effect. Lactate increase remains. And blockers of beta-2 adrenergic receptors, ICI, this 118551, and this completely block the induction of lactate. So it is beta-2 receptors and not beta-1 receptors that are required for lactate release in the hippocampus. And these are the memory of this animal tested. And we see a parallel effect. The animals have been injected systemically. So propranolol blocks, beta-1 does not, and beta-2 blocks long-term memory. So it is beta-2 that is involved in this memory formation as well as lactate release. So we're focusing now on beta-2 receptors, and also look at if we inject beta-2 blockers directly in the hippocampi, do we see the same effects? And the answer is yes. If we inject ICI, memory is impaired, the effect is sustained. We give a reminder shock to see whether we can rescue the memory, but it's not. Whereas the betaxolol, the beta-1 blocker, has no effect. So again, confirming that it's beta-2. And the next question was, well, let's see what is the distribution of these receptors. So she did this autoradiography and look at binding of beta-1 and beta-2 adrenergic receptors. And she found that obviously they have a different distribution. This confirms previous data. We didn't discover that. And this distribution is interesting because if we look now, we zoom in the hippocampus, we see that the beta-2 adrenergic receptors are particularly enriched in the oriens and in the lacunosum molecolare. Beta-1 is more distributed evenly. And this distribution actually corresponds to an enrichment of astrocytes in those subregions. So beta-2 are most likely enriched on astrocytes. And again, there is some suggestion in the literature that says beta-2 adrenergic receptors are mostly on astrocytes and not neurons. So she went on and then decided to knock down selectively the expression of beta-2 either on astrocytes, in astrocytes, or neurons, using viruses that express sequences that will knock down the expression of beta-2 receptor, so shRNA silencing, or antisenses, under the specific promoter that are expressed either in astrocytes or neuron selectively. So this is an example of the virus that she used. This uses the GFAP promoter, so it's an astrocytic promoter. And in fact, what she sees is that the expression of this virus is selectively in astrocytes and not in neurons. You see, with the immunostaining, the green is GFP, which is the virus expression. The red in this case is NUN, a marker of neurons. And when we do the merging, we see that they are not in neurons. And in the previous one, there was, there we go, merging of the staining, so it's an astrocytic expression. So using this virus and targeting selectively astrocytes, she confirmed that the beta-2 adrenergic receptor expression is knocked down by the viral infection, but not the beta-1 adrenergic receptor levels. And then she looked at memory. And what she found is that these viruses that knock down beta-2 adrenergic receptor in astrocytes and not in neurons lead to memory impairment. Again, the effect is sustained. A reminder shock, no effect. Now, when she gives lactate, in the scramble, in the control, she sees no effect. When she gives lactate with the silencing, the shRNA against the beta-2, she completely rescues the effect, the memory impairment. So she went on and then asked, well, what if we target beta-2 energy receptors in neurons? So she engineered another virus. Now the promoter is synapsin that targets neurons, and in fact the virus is targeting neurons selectively. This is the merging and the co-staining with the neuronal marker. No co-staining with astrocytic markers. Downregulation of beta-2 adrenergic receptor expression in the hippocampal extract, not of beta-1. As you can see here, the downregulation is much smaller, in agreement with the idea that beta-2 adrenergic receptor in neurons is actually less, the expression of these receptors. And then she looked at memory effect and the latency of these animals infected with this virus, where the beta-2 adrenergic receptor in neurons is knocked down, had no effect. Memory was comparable to controls. Memory was strong. She tried many different conditions, changing the weeks of infection, therefore the persistence of this silencing. She changed also the shock intensity. This is 4 weeks after infection. This is with 2 weeks and 4 weeks with 0.6 milliamps, so lowering the shock intensity to see if she could see any effect. Thus far, we have not seen any effect by targeting by beta-2 adrenergic receptors in neurons. So in conclusion, the effect of hippocampal beta-2 adrenergic receptors on memory formation is astrocytic rather than neuronal. Noradrenaline via beta-2 adrenergic receptors on astrocytes mediate lactate release, long-term memory, and the underlying molecular changes, including those occurring in neurons for long-term memory formation. So in conclusion, glycogenolysis and astrocyte neuronal lactate shuttling are required for long-term memory formation, and stress, noradrenaline-mediated memory consolidation and modulation. And in summary, identifying the metabolic needs, regulation, and optimal energy and nutrition supplies for a healthy brain or disease throughout the phases of life is greatly important. Chocolate is an antioxidant, will help. Thank you, Switzerland and Nestlé, for that. But many thanks who actually did the work. I already mentioned Virginia Gao, Akinobu Suzuki, and Sarah Stern. Great collaborators, Pierre, of course, Sylvain, who helped in the viral constructions, George Huntley and Özlem Bozdaji at Mount Sinai and at the VA that basically taught us how to do microdialysis in vivo. Obviously, the funding, the National Institute of Mental Health and MedNight. And this is the current lab, and I need to add to the thanks also Gabriella Polonini and Michael Steinman, who helped in this project. Thank you very much.
Thank you, Christina. We have time for some questions. Yes?
Very nice, over here.
Well,
Very nice presentation, thank you. I see a lot of allergy of similar type of biochemical pathways like you see in muscle metabolism, glucose metabolism, glycolytic, oxidative, also glycogen storage, but I didn't hear anything about lipid metabolism. So I'm sure there must be also a big role for lipid metabolism in the astrocyte-neuron interaction.
I totally agree. I totally agree. And that's why in the introduction I try to say metabolisms, because I'm sure, I think we really know very little about it. So, and I hope this is the beginning for opening a number of questions and understanding how metabolism works. Particularly interesting is in activity-dependent processes, right? Yeah.
Question there? Oh, thank you. Yeah. Oh, sorry.
Okay. As I'm not so much into this, I'm kind of...
You don't want to introduce yourself?
Sorry, I forgot. Nedergaard from Stockholm. Not working directly with these things. So in order to... I try to think how the rat will think, and then I think like I will do myself. And then I start to wonder, is this really memory? Because if I walked, let's say, in here, and got an electric shock, then of course I will stay out of here. However, if some time passed and I came here again, a little thing in me may say, not that I've forgotten the shock, but there's another thing we could call a curiosity drive,
Sure.
which means that after some time I may start to actually go in and just to be sure that I actually will get a shock again. So can one distinguish between this as a memory and a balance between curiosity, which rats are, and...
Wonderful question. Yes, I mean, there are a number of experiments that do that, or at least attempt to do as much as possible. The first thing I want to say is that there is individual variability in humans as in rats. So some of them, let's say with 0.6 milliampere shock, it's a mild foot shock, one single event, they don't go in for a very long time. You know, we can wait 18 minutes and they don't want to go in. And other rats, they wait, they have a latency, but then they go in. So first of all, there is individual variability, which will be interesting to understand why. Maybe, you know, this curiosity to enter again. And the second thing is, what we are interested in the lab is exactly what you are saying. So we run a number of controls. And for example, is it anxiety? Is it that, you know, they don't move because they are anxious, but is it memory or anxiety? So that can be dissected with a number of behaviors and anxiety tests, for example. And we see that anxiety tests are not changing responses, yet they have a latency. So with a number of tests, we can conclude with some certainty that is more the association and the memory of it rather than something else that is more controlling the expression of the behavior. But the third thing I want to say is that we are interested very much in this. Time is one important variable. And this ensures that the brain remains healthy. So if it is a single event, what happens over time is that the memory first strengthens without doing anything, just time. I mean, something is happening in the brain, clearly. We can talk about that. Perhaps it's the reactivation of the trace, maybe implicit reactivation, sleep, and other type of explicit reactivation as well. Remembering what happens. If some aversive experience has occurred, in the time after the experience, we keep thinking about it. So that's a reactivation, and we are studying very much the reactivation of the memory, retrieval of the memory, and what does to the memory trace. And it's interesting that in the first temporal window, after the experience, retrieving the memory makes the memory changeable. So memory, as I mentioned, is labile initially, and then becomes stable, resilient to interferences. And the first phase is gene expression dependent. So if we wait a few days and we interfere with gene expression, memory is fine. It's not going to be disrupted anymore. And I'm talking about hippocampus now. So that memory is considered to be stable. But retrieval makes the memory labile again, and again for a limited temporal window, which is an interesting temporal window of opportunity, because in that temporal window, we can modulate memory. We can strengthen it, we can weaken it. So for a clinical application, it's very interesting. What happens with time? If we wait several weeks and we retrieve the memory does not become labile anymore. But it becomes more prone to extinction, meaning if we expose to the context without the shock, they actually diminish the avoidance response and they enter into the dark side more quickly, exactly what you are saying. So that's why I'm saying this is an interesting process that keeps ourselves sane. So one aversive experience should not lead to PTSD. So with time, the changes that occur in the brain lead that memory to weaken, to change the quality, and to be more sensitive to extinction, and therefore then to explore other places that may remind us of an aversive experience.
Okay, we have several questions.
Sorry, I'm going to keep it brief, but this was interesting.
keep them short. Tamás, and then
I'm
Bruno.
glad that you mentioned.
Yeah, but I know it's not to be the wrong.
Is there an administrative fitting order?
No.
Can you introduce yourself?
As you hear from the Hungarian accent, I am Tamás Bárta. I am glad that you mentioned post-traumatic stress disorder, because we tested 8,000 compounds which crossed the blood-brain barrier to be given to soldiers who were exposed to events which were most likely to lead to post-traumatic stress disorder. The only compound which stood out was propranolol, but the discussion was always, so it is given if the cardiovascular state permits of the soldier. It is not. But it was always questioned whether it has to do with that it is almost the only really effective agent to reduce heart rate.
Yes.
And your experiments are wonderful in the sense that you engineered viral knockdown directly in the brain. However, I wonder if you have reached ceiling effect. So if you combine propranolol with the astrocytic lactate transporter blockade, would that be an additional effect, or you are already at the ceiling? This is a very important issue because propranolol is far from perfect, and this is a very big problem. And I have a comment. When you talk about IGF-2, as you know, in Stockholm, because of Kerstin Hall, a lot of work has been done on IGF-1 and insulin. And in that work, we also have, of course, worked with IGF-2. And as you know, the insulin receptor is a dimer, and it can bind simultaneously insulin and IGF-2, or insulin and IGF-1. And you ought to examine whether heterodimeric occupancy by the two agonists, which is fully possible, dependent on the relative concentration, and insulin concentration in the brain varies, has a different effect than just having a pharmacological dose of IGF-2 when you surely will have a homodimeric coverage of the dimeric receptor.
Oh, absolutely. Two wonderful questions. I'm going to start with IGF-2 to be more brief. The other is a little longer, my answer. So with IGF-2, what we did was to test whether IGF-1 receptor or IGF-2 receptors are mediating the enhancement, and we found, so these are blockers of the homodimers, IGF-2 or IGF-1. And we found that IGF-1 receptor is not involved. It's IGF-2 receptor that mediates memory enhancement. We didn't test the hybrids yet, but definitely IGF-1 receptor had no effect. And those receptors are very different. Insulin and IGF-1 receptors lead to growth, and IGF-2 receptor leads to lysosomal degradation. Very different. Cellular.
Not in binding, not in forming heterodimer.
Right. If in the heterodimer we didn't test yet, but we know there is a distinction between those homodimers. For the propranolol, we have worked extensively on that because especially with reconsolidation, memory retrieval, giving the opportunity to disrupt the memories again. This was thought to be a wonderful potential intervention for PTSD as well as addiction. So we retrieve the memory of the trauma, memory becomes labile, we block reconsolidation, and things are going to be good. And propranolol has been shown to block reconsolidation with Pavlovian fear conditioning. When we tried it in inhibitory avoidance, there was no effect. So this is not all bad news, because perhaps reconsolidation is actually working with the amygdala modulation. And perhaps there could be compounds or intervention that will decrease the emotional re-elaboration of the memory, not the content, not the hippocampal dependent content. We don't know that, and we are setting out experiments to test that. But with inhibitory avoidance, propranolol injection systemically, we have done a lot of studies with that, no effect. What had effect is glucocorticoid receptors blockers. RU486, known also as mifepristone. So that is another pathway because it's not clearly, right? But more studies need to be done. With the lactate metabolism, we didn't look at reconsolidation yet. So there could be a number of potential questions that are interesting. And lastly, studying all these compounds on fear conditioning tasks that are adaptive is not the answer. Memories of PTSD are not of the same quality of these memories, right? We all studied that because we wanted to look at reconsolidation effects. But the more I study that, the more I know that the memories that are pathological, like those of the inverted U, are not of the same quality. So we cannot test the compounds on normal fear conditioning memories. We have to develop PTSD type of models. And we are doing that. We actually have an interest.
The military protocol doesn't fall for only the recurrence of PTSD. But I was referring to the acute. When you pull out somebody of a tank where everybody else is dead, we give now propanolol. Question is, would propanolol plus the astrocytic lactate transporter inhibitor be synergistic in that when you form what becomes the PTSD?
Sure, we have to test that. We don't know the effect we see. They are very strong. But the second thing is they probably have to be given together with activity. So we cannot just give propranolol weeks later, right? We need to have the memory trace reactivated, and that's why the reconsolidation protocol is interesting. But I agree with you, for prevention, not after when memory is consolidated and it has a different quality.
Bruno Vellas from the Nestlé Nutrition Council. You have shown to us the impact of lactate, noradrenergic, and cortisol on long-term memory and emotional memory. But the patients that we see every day, they have short-term memory problem for usual life events, like what they have done yesterday, what they have to do today. Do you think that that will work all over?
What patients are you talking about?
The patients that have a memory problem, older adult, or Alzheimer's disease?
Alzheimer.
They have mostly short-term memory for small events.
Right. So we didn't test yet these effects on short-term memory, but there is a paper from Paul Gold's lab looking at working memory. So it's a short-term memory effect, again targeting the hippocampus though. So there is a lot to be done, right? Because working memory in humans is more cortical. So we need to know more about region specificity, time, short-term versus long-term, and so on. But there is this paper suggesting that working memory is actually blocked if glycogenolysis is blocked.
Question here?
So Johannes Bench from Nestlé. I wonder whether we need to distinguish between lactate and lactic acid. So only in the undissociated form, I know this from microorganisms, from biotransformation, only as the acid, the molecule can penetrate the cell wall. Inside the cell, it dissociates, and then you have a pH effect. And this stimulates in microorganism activity. So you need to have a certain concentration, and this increases the activity. If you exceed this concentration, the external lactate or lactic acid concentration, then the cell breaks down. And this is what we are using in food industry actually as a preservative. You can use lactic acid as a preservative. In low concentration, it stimulates. In high concentration, it inhibits. So we need to distinguish clearly between lactic acid and lactate.
Wonderful. Yes, we didn't do any experiment, but yeah, absolutely. It's a wonderful suggestion. We'll do. We know that the non-active, the D-lactate, that blocks, but yeah, we should do that. Yeah.
I mean, one I can say is that the transport of lactate through the transporters is with protons, so it's lactic acid.
Yeah, so the lactic acid is going through the cell wall, and then inside it dissociates. And this stimulates the cell because it needs to get the proton out again. But if the concentration is too high, the cell collapses.
Yeah, we didn't use high concentration, so well, I mean it's an easy experiment to do.
Question here?
Thank you. I very much enjoyed your talk and learned a lot as a nutritionist, not in the neuroscience side of things. It struck me that there's a second approach you could take because your initial effect is a shock and survival comes in there. Suppose the rats entered and enjoyed it and had a good time and then you deny them access to it. Do people work in that realm?
Sure. There are tasks designed
And
that way.
Does it come out the same way? Are the results the same way?
We didn't test that yet. We didn't test that yet. We need to test different types of memory, different types of memory systems, the valence. Clearly, if there is an emotional valence, that's going to be an interesting set of questions. I don't know of anybody, I mean, condition place preference has been actually studied. So that is a memory evoked by a drug. Did you use cocaine, right?
It was cocaine.
So, and Pierre's collaborator.
Block the same memory, but with DAB blocking glycogenolysis with cocaine preference.
So it's not only aversive memories. But I agree that different types of memories needs to be tested to see what is the requirement.
1, 2, 3, 4, and then we stop. Otherwise we have to skip minutes.
Yeah.
So
Raj Ratan, Burke and Cornell, very nice talk. I'm curious what's different about generating glucose 6-phosphate from glycogen as opposed to glycolysis. Why doesn't D-glucose rescue the norepinephrine effect, given that fact that they're both going into the same pathway?
Yeah, $1 million question. I don't know if Pierre wants to take it, but I'll give you my answer, and then Pierre can add to that. So this suggests that lactate does something different than glucose, right? And as we have seen, glucose enters into astrocytes because from the blood. Astrocytes are connected to the blood. So the glucose is entering astrocytes, and there is going to go glycolytically, or it can form glycogen. I mean, at one point in time, there's going to be the restoring of the glycogen stores, probably during sleep. There is literature suggesting that, so that we have enough then to be used. And then lactate transported into neurons is going to be supplying the energy to also avoid the high oxidative metabolism that will occur in neurons. And as I mentioned, too much oxidation will lead to damage. So the lactate, instead of glucose, will also help in redox modulation and in ensuring that the homeostasis is kept for neurons to work well and in a prolonged state. Because when this high energy demand is required, the neuron itself cannot be used. So why glucose versus glycogen? They all are entering through astrocytes. And one can think that are both used when there is an activity-dependent process. High energy demand, we're going to call glucose in and use glycolysis. We're also going to use the glycogen storage, depending on how much energy is required. And then lactate formation, and lactate is going to supply energy and something else for homeostasis to neurons.
I think we can really just stop now. I think otherwise we'll be over time. I'm sorry. Christina is here. You can ask her questions.
Thank you.
Kuzawa from Northwestern University in Evanston, and he will talk about building the costly human brain, implications for the evolution of soul childhood growth, and the origins of diabetes. Chris?
Well, it's wonderful to be here. I really appreciate the invitation, and I've learned a lot, and like many of the speakers before me, I'm neither a neurologist or a nutritionist. I'm a biological anthropologist, and actually my work generally has very little to do with the brain. Most of what I do is in the Philippines. I work with a study in a place called Cebu, where there's a birth cohort that's been going for 32 years, tracking mothers, their offspring, and their grandoffspring. So more than 3 decades of data. We use these data to look at the long-term effects of early environments, stuff related to what Steve Matthews was talking about yesterday. We also look at aging, genetics, epigenetics, all sorts of things. But I'm not going to talk about any of that today. I'm going to talk about a different question that I've been thinking about and writing about for about 20 years since graduate school. And that is, how did humans manage to evolve such large and energetically costly brains? And this is particularly an important question early in life because the brain is much larger. You know, we've heard this figure batted around that about 25% of resting metabolism goes to the brain. But as I'm going to show you, that number is much, much greater during childhood. And I think this has implications for understanding the evolution of the human pattern of growth in our life cycle. And it also will have connections with things like cognitive development and also possibly the origins of diabetes. So I'm going to start with a short preamble because I am going to come back to this point about diabetes at the end. I just want to set up some of the literature that sort of motivated that question. We already heard about some of this, and I think many of you are familiar with it. This is the work of David Barker, showing that if you're born low birth weight or lower birth weight, you have a higher risk for cardiovascular disease. And he proposed this model, which I think has stood the test of time pretty well at this point. A fetus that's undernourished or stressed is going to slow its growth rate, but also modify all these other biological systems in ways that are relatively permanent or durable and persistent to later life to influence your long-term risk for chronic disease. And some of this is epigenetic, and some of this involves growth of organs and so on. So this is pretty well established at this point. I think many people are aware of it. But these processes do not end at birth. They continue in the postnatal period. In fact, there's a lot of evidence that if you're born small, these long-term health consequences are actually much worse if you manage to grow quickly after birth. And so the general finding, like I said, being born small, you end up gaining more visceral fat, having insulin resistance, among other traits. And so just a couple quick examples of that. This is the ALSPAC study in the UK. Individuals who experience catch-up growth in the first 3 years of life are more insulin resistant. They have less insulin sensitivity at 8 years of age. Now, if you're less insulin sensitive, that means that your muscle and your fat are not responding to insulin and not taking glucose out of the bloodstream, and therefore you're at increased risk for diabetes. And this is something very similar from Chile. So rapid growth in the first 3 years of life, you see more insulin resistance, okay? Now, we can dig into this and try to figure out what the mechanisms are at a cellular level, at a hormonal level, organismal level. But the question I want to pose today is a little different one. From an evolutionary perspective, why does growth in infancy and childhood seem to matter? Why does our body reset its metabolism, its priorities with where glucose is used within the body in response to the nutrition that we experience during this period? And I'm going to tell you right now that I don't have a great answer for this. I don't have the definitive answer. But I do want to convince you by the end that if we want to have a full understanding of this, we're going to have to take the brain seriously. Because the brain, as we've heard, is a major user of glucose, and as I will show, is really dominating the body's metabolism at this age. Right, and so to set this up, go ahead and dig into a number of topics. We're going to start by looking at the evolution of human child growth. There's some very unique characteristics of growth in humans that differentiate us from other species. And then we'll talk about some of this research that I and my collaborators have done in recent years that quantifies the cost of the brain and looks at that developmentally. And then we'll have a few implications for international nutrition at the end. Okay, so let's start with this. Now, this figure compares us to chimpanzees. Now, chimpanzees, our last common ancestor with chimps was about 6 million years ago. They're our closest living relative, fairly closely related to us. Now, what you see here, there's some obvious differences. Chimpanzees, like other mammals, okay, they wean their young at the age at which their offspring can fend for themselves and feed themselves, which is about 4.5 years of age. So chimps go from breast milk to self-provisioning, and it's, there's really no food per se that's provisioned. Of course, our offspring, anybody who's had a kid knows that they're not going to be able to feed themselves until quite a bit later than that, probably 5, 6, or even later, right? And yet we wean much earlier than chimps. So how do we pull this off? We do this by for instance, making baby foods, weaning foods, right? Processed foods. And the other key factor is we are what is called a cooperatively breeding species, in the sense that it's not just the mother that has to feed the baby, it's also the father, grandparents, older siblings, other individuals, right? Many people end up feeding the child, so the burden is not just on the mother. Now, what this allows us to do as a species, it allows us to wean early, which means ovulation resumes earlier, which means that there's a shorter interbirth interval and fertility is higher. So to evolutionary biologists, this is kind of a no-brainer why this system would actually persist, right? Because there's a clear evolutionary advantage to it. But the other piece of this story that you can see is the fact that the blue line is so much longer, right? We also delay the onset of adulthood. We continue growing much longer. And so this piece has been less well understood. Why is it that our growth slows so much? And if you think about it, we grow for much longer, and we don't end up to be the size of elephants, right? I mean, we end up about the same size as chimpanzees, so clearly our growth rate must be a lot slower. So let's take a look at just how slow our growth is. And I'm going to start with this. Many of you know this. This is the Kleiber line relating metabolic rate to body size across different species. And you'll see that both of these scales are log-log plots, and the exponent here is about 0.75, so it's less than 1. And what this means is that as you go from small to larger-bodied organisms, metabolic rate is, of course, increasing. The caloric expenditure is increasing, but it's increasing at a slower rate than the gain in body weight, right? So a gram of elephant uses about 1/8 the calories of a gram of mouse, that kind of idea. There's an increase in efficiency. Now, given that metabolic rate scales in this way, okay, to body mass, it makes sense that other components of our metabolic expenditure also scale this way, and we see this with growth rate. So these are data compiled by Case some decades ago, showing mammals and reptiles. And the first thing, and this is growth rate between birth and about 40% of adult size, so it's roughly the time frame of childhood that we're thinking about. First thing I want you to notice, the scaling coefficient is about the same, right? So it agrees with that Kleiber line. The second thing is that if you look at mice, rats, guinea pigs, and sheep, okay, major experimental animals, they're right on that line. They're very well-behaved mammals. They look good. What also makes sense is down below you have the reptiles. Now they're cold-blooded, right? So for any given body mass, they expend much lower calories than a comparably sized mammal, and so naturally they have less calories to expend on growth, and they have slower growth. So that all makes sense. What doesn't make sense is why humans are on the reptile line. Okay? This is where the story gets kind of odd. So if we take this line, if we just look at these species that are right in our body size here, and plot them in order, okay, these are the mammals that are of our size, and you can barely even see the human line, right? It's orders of magnitude slower than these other species. And if you compare us to reptiles, we look like a python. So there's something very strange going on here, right? Humans grow at the pace of a reptile during childhood. Why is this? Now, one idea that's been cited in the anthropological literature for a long time is that building the human brain is unusually costly, and this has required a slowing of growth. And this is just kind of an intuitive idea that people have batted around. It's never really been tested, but it's cited a fair amount as one plausible idea. And the problem is, how do you go about testing it? Well, first of all, here you see the increase in brain size over about 4 million years in our species, right? So we're very encephalized. There's been an increase in brain size of maybe a factor of 3 or 4 during that period. And primates as a whole, compared to other mammals, are very large-brained. And this is just limited to the apes, which are even larger brained, and we're clearly at the very top of the heap, right? So we have very large brains, and they are costly, as we've heard about. So if you compare a gram of muscle tissue in the adult to a gram of brain, it's about 15 times more costly, right? And so all of you are sitting here at rest, and your bodies are giving off energy at about the rate of 100 watts. So you're heating up the room at that rate. 20 of those watts is your brain, and that may seem kind of dim, it's not particularly bright, but it's actually pretty remarkable, because if you look at other species, most are in the order of about 2 to 3% of their metabolism goes to the brain, right? So we're different. Other primates for which there are data are around 10%, and then the human, again, is about 20%, as we've heard. Now, naturally, okay, so here's the adult. In the newborn, the brain is much larger relative to the body, so this figure is going to be much higher. And the old estimates from the literature had that figure at 60%. Okay? So early in life, the brain really is dominating the body's energy budget in a way that we can imagine might actually lead to a trade-off with growth rate. Maybe some of the calories that go to growth need to subsidize our brains. Now, if that's true, then we can come up with a hypothesis and some ideas that we can test. So if our costly brains explain our slow growth, the ages of slowest body growth should also be the ages of highest brain energy needs, right? The ages when the brain is really demanding a lot, we should see growth slowing down a lot. And so we can look at this again by looking at percentage of RMR going to the brain. If you think of the body's energy budget as a pie, what fraction of that pie is being dominated and used by the brain? How much is left over for other functions? This tells us something about how demanding the brain is for the body at that age. Now the problem is that we don't know a lot about that until recently. We don't really know developmentally how that figure of percentage of RMR to the brain changes. Now one approach that's been taken is to take the expenditure per gram of brain and multiply it by the size of the brain at different ages and then divide by the resting metabolic rate. And this is in fact what was done by Holliday. Took the expenditure of the adult brain and then multiplied it through and then divided by RMR. And this is the data, kind of connected here to show a graph. And this figure has been cited a lot. It came up with this estimate that 87% of RMR goes to the brain at birth. Now, I've always had problems with that figure. That doesn't seem plausible to me, because that leaves 13% of the body's metabolism to do everything else, the heart and so on, the organs. So it always seemed like an overestimate. And also, if this is in fact the nature of that developmental change in brain energetics, then there's no evidence for a trade-off between growth rate and brain metabolism, because the age when body growth rate is slowest, which is in childhood, about 4 to 5 years of age, is not an age when the brain is at its peak. If anything, it's kind of midway down and declining, right? So if anything, these graphs are more parallel than suggesting a trade-off, right? Now, the problem with this is that this assumption that a gram of brain costs the same at different ages is wrong, and that's not surprising because there is a lot of activity going on early in life and that we've already heard about. And we get insights into this from PET imaging, positron emission tomography. This is a method, this is not something I do, but a collaborator does, where you give a radio-labeled glucose, right? It's a tracer, and then you can measure the rate of glucose uptake in the brain. You get this kind of heat map that tells you about the rate of energy use. And Harry Chugani, back in 1987, published this classic paper where he compiled PET data from his clinic on kids varying in age from birth to adulthood. And here's some adult controls. I put a red line through there for comparison. And what you see is that each gram of brain, and this is cortical, uses glucose at a slower rate than the adult at birth, And that it actually increases a lot to about twice that level in childhood, and then it gradually declines, right? So there's a lot of dynamics in the rate of glucose uptake per unit gram of brain. So what underlies this? Well, this is all largely about synaptogenesis, right? And we've heard a lot about how costly synapses are. Part of the way the brain develops is proliferating extra synapses. And then when you use your arm or you speak or use different parts of your brain, you're actually firing those circuits and they stabilize. And the circuit in the synapses that don't get fired eventually get pruned away. So you get this initial proliferation and the pruning away, right? And this is a costly process. And you can see early on there's skills related to just hearing and sight, and then there's language after that, and then you get higher cognitive functions that are spread out over a longer period. And we've already heard much more about synapses and their energy metabolism, but the bottom line is that they have constant requirements for energy. You need ATP to maintain the gradients of potassium and calcium, and sodium rather, and which allow neuronal firing. And so here you see synaptogenesis. This is the actual synaptic densities. There's not many data on this. This is a classic paper from 1979 by Huttenlocher. You see the rise of synaptic density in the early years, peaking out in childhood, big gap in data, but eventually getting down to adult levels later, right? And so this is synaptogenesis, and this is the process of pruning that I just talked about. So this is largely what underlies these dynamics. This is really reflecting changes in the density of synapses, which are energetically costly. what this all means is that this figure is going to have to be revised, right? At birth, this is an overestimate because, as I showed, each gram of brain actually has a lower metabolic rate at birth than in the adult, right? So that's an overestimate. But as I noted, it rises to about twice the adult level in childhood, so this is an underestimate. And we kind of hypothesize that the curve might look something like this once you take all that into account. And so that leads me to the second point of my talk, which is our recent study that tried to pull together data and quantify all of this. And these are some of my colleagues, my collaborators on this from Northwestern, Wayne State, George Washington University, Harvard, and Mount Sinai Hospital. Okay, so we started really quickly. This is a very truncated overview of the methods. We started with Chugani's data, PET data that I already showed you. We used the cerebrum, cerebellum, and brainstem, set separate curves to these, and these collectively give us the glucose uptake by the entire brain. That gives you the energetic cost per gram of tissue. So we also need to know how many grams there are, and we got that from MRI data. There's an NIH longitudinal study that we mostly used, and Nick Lang, who was part of that study, was also on this paper. And then you do all the math, and you get total brain costs. You do that for each age, right? And this is what you get. So this is what it looks like. Now, we ended at 15 years of age because that was the last PET data. And I've got the adult level down here. Now, grams of glucose per day is peaking out at about 5 years of age. There's about a 10% difference here. That's because males are about 10% larger, both their bodies and brains. So that's all that's going on there. But what's remarkable about this is that at 5 years of age, the 5-year-old's brain is using twice the glucose of an adult's brain. And this is remarkable because their brains aren't even full size yet, and more importantly, their bodies are about 1/3 the size. So as a burden on the body, this is really significant, right? And so we want to be able to quantify that. And so the next step is to convert these grams of glucose into kind of caloric equivalents and then divide by resting metabolic rate to get a sense for what percentage of the body's RMR is going to the brain, like I showed earlier, and this is what those figures look like. It's peaking out at about 2/3 of RMR at about 5 years of age. Right, so this is really substantial, right? It's really dominating the body's energy budget. And so this leads to a question, right? This gets to the point that I set out to address. Are ages of high brain glucose use also ages of slow weight gain and vice versa? And the answer is yes. So let's take a look at it. So here, this is weight velocity calculated just as the first derivative of this cross-sectional data that we're using in the brain study, so the body sizes of the same individuals. And it's, so it's a crude estimate, but it gives us a sense for a kind of inferred weight velocity in the sample. Converted it to SD units, standard deviation units, so it's unitless. And then you impose upon that the glucose curve. And this is what it looks like. So the age at which glucose use is at its maximum is the age at which body weight growth is at a minimum. Now, we can go one step further than this. And instead of plotting these both versus age, we can plot one versus the other. And if there's a trade-off between them, we should expect an inverse linear relationship between the two, right? As the brain becomes more costly, growth rate should go down and vice versa. When we plot them that way, this is what we see. And if this was a perfect inverse linear relationship, it would be that gray dotted line. So I think we're pretty close. So let's take a look at what's going on here. So each of these is a different age, and we're kind of tracing through that curve. So let's go between 1 and 2 years of age. As you go from 1 to 2, the brain is increasing its dominance of the body's energy budget. This is partly because of growth and partly because of synaptogenesis, right? Both of those processes are happening. So that's increasing. At the same time, weight velocity is decreasing, right? And you'll notice that each is about 1 standard deviation unit, meaning that they're proportionate. So there are proportionate inverse changes happening in each. Here you get to this point, and this is the point where the brain is at its maximum in terms of dominating the body's energy budget, right? It's the rightmost point, and it's also the lowest growth velocity, right? So this is the point where the peak in brain energetics coincides with the trough in weight gain, and on the other side, we reverse this, right? So from 7 to 8 years of age, the brain is starting to decrease in its dominance of the body. This is because of synaptic pruning, and at the same time, you have about a proportionate increase in weight velocity. So I think we have really strong evidence here that there's been a trade-off between these. And this is what it looks like in females. It's similar, but puberty happens earlier, so things change at an earlier age. Okay? Okay. Wow, it's only 20 minutes in. I'm going quick. We're going to have a lot of time for discussion. So let's go back and where I began here. Humans have this very unusual life cycle where we really slow our growth rate, and I think we've got some good insights into why. Our brains are incredibly costly. So costly that some of the calories that would have been going to growth are actually going to the brain, right? And so I'm going to dig into a couple of the implications of this for international nutrition. And one, coming back to this point about diabetes, and then we'll talk a little bit about cognitive development, which I'm sure the next speaker is going to have much more to say about. All right, so we showed this graph showing this evidence that effectively calories are going from body growth to glucose, right, at this age of very high brain requirements. One implication of that is that fat deposition grinds to a halt. Right, so this is, all I did here was take some kind of reference data on body composition, multiply it through by the body weights in the sample, and then get an estimate of fat mass, and take a derivative of that to tell you kind of the velocity of fat deposition and how that's changing. So at birth, there's about 3 kilograms per year if they were to keep growing at that pace. Okay, that's how much fat is being deposited. But by the age of the peak in the brain energetics, it's down to about 10 grams per year, right? So it basically comes to a halt. And this is interesting because it gives us some insights into some other patterns that we know about. The fact that this shows skin fold levels, right? So adiposity and BMI bottom out at about the same age that the brain is at its peak. Right, now this is interesting because this suggests that the brain's needs are peaking out at an age when we have our lowest energy reserves. Now this is important because unlike other tissues in the body, the brain's metabolic rate is relatively stable. Even when you're sleeping, it's still using a lot of energy. And if you're under nutritional stress or under starvation, for instance, you still have to meet those basic energy needs, right? And so there's an interesting dilemma here, and you can think about this with an analogy. I mean, many of you have mortgages on your houses, and you have to pay that mortgage every month. And if you lose your job, you still have to pay the mortgage or you have to foreclose on the mortgage, right? So you better have something in savings to pay it. Now, if you have a bigger mortgage, you're going to need to have a bigger savings account in order to make that mortgage payment and avoid foreclosure. It's completely analogous to what we have here. Because during childhood, 66% of the child's income is going to a mortgage, right? And that means that if there's any kind of event that disrupts their intake, they'd better have a sizable savings account to draw from. But in fact, it's at its lowest point in the life cycle. So we have a bit of a paradox here. How can we have this very precarious situation? And I think that this gives us some insights potentially into some of the things that I set up at the beginning, okay, relating growth and diabetes. So as a reminder, fetal growth restriction followed by rapid postnatal weight gain leads to the following. Okay, weight gain is favored, as you gain weight, favors fat over lean, so you end up gaining more fat. That fat is preferentially deposited in the visceral depot. And that's important because the visceral depot is actually different than other depots in the body. It's innervated by sympathetic nerve fibers. So when you experience stress and adrenaline, part of what's going on is adrenaline's being released in that depot and fat is being released immediately. So this depot, unlike other depots, is actually part of kind of short-term energy balance regulation, much more so. So that's interesting. And then muscle, as I've already pointed out, is insulin resistant, right? And so if we think about this, of course these contribute to diabetes, but thinking about it functionally, we might think of these as ways for the body to buffer the brain. And so let's take a quick look at how that might work. So here we have body fat, liver, muscle, and brain. And if you experience starvation or some other stressor that causes you to break down triglycerides, you have direct buffering of the brain. Okay, glycerol enters the liver, is a gluconeogenic substrate, becomes glucose, and feeds the brain directly. You also have free fatty acids, which go to the liver and lead to the formation of ketones that are taken up by the brain preferentially and used as energy substrate. Okay, so you have direct buffering of the brain through fat. But you also have indirect buffering, and this is where insulin resistance comes in. When free fatty acids are in the bloodstream, muscle becomes insulin resistant. It stops using glucose and preferentially uses the fatty acids for substrate. And this is a way of in a sense, conserving glucose for more important functions. this increases the amount of glucose available to the brain indirectly, right? And so you can think about it this way, this schematic here. If insulin resistance reduces peripheral glucose uptake, then it leaves more for the brain, which does not require insulin for its uptake, all right? And it's one of the main non-insulin dependent users of glucose. So we can think of insulin as having this function. As you know, of course it leads to diabetes, we know that. But from a functional perspective, it's really about the body reprioritizing where it's using energy and moving it from less vital functions to more vital functions like the brain. And if that is what its function is, then this age here this is really where I think where some of the action is and where evolution must have shaped these responses, because this is where 66% of our income has to go to that mortgage that we have to meet each month. Okay. Now, I'm not going to have a lot to say about this because this is outside my area. I just want to have a few speculations at the end. Implications for the role of nutrition in cognitive development. And there's been a lot done on this topic. And here we see the brain growth velocity curve. So very fast brain growth here, slowing down. And so naturally, a lot of the focus has been on this period of rapid brain growth. Okay, because it's not only the point at which the brain is growing fastest, but it's also a period when you have weaning, infectious disease, growth faltering, all right? It's also a critical period in body growth, so stature growth. Most growth stunting has occurred by 2 to 3 years of age. So there's a lot of action here in terms of nutritional stress, and much of the work on the role of malnutrition in cognitive development has focused on this period and has found pretty strong evidence in favor of it. And I'm not going to deny that, but I think that our work points to another potentially interesting period. Okay, this is a period when the growth of the brain is mostly ceased, at least towards the end here, and yet you have this peak in brain energetics. And so in what ways might nutritional stress impact some of the processes that are being laid down during this period? The studies that I've seen, and I'm not an expert in this literature, most of them find strong evidence for here, not as much here, but a lot of those studies actually just focus on here. So I think that there's enough of a question here that maybe some additional attention needs to be focused on this. And it's a very simple kind of starting hypothesis. Oh, and here, again, I already showed you this graph, but this shows you that what's developing at this age are higher cognitive functions, right? So things like executive function. So we might imagine that those would be impacted by nutritional stress during this period. So here's some two very simple and not mutually exclusive predictions. That chronic or severe energy stress, including infections, during the brain energetics peak are going to lead to long-term deficits in the higher cognitive functions that are developing most rapidly at that age, and or developmental delays that work to protect those cognitive outcomes, but possibly with other ramifications, social ramifications perhaps. So these are just a couple of the reasons that I think we need to take the brain and its energetic costs during development very seriously. I think it has implications for a whole range of different questions. And with that, I will acknowledge my co-authors and funding agencies, and thank you for your time. Sorry.
Thank you very much. Richard?
Very short, so yeah, we have a lot of time, I guess.
Richard, can you introduce yourself?
Richard Frikovic from Paris. So if you just go back one slide, if
Sure.
that's possible.
This one here?
So I thought it was a fascinating bringing together information from lots of places and constructing a wonderful overview and theory. But is it necessarily so that in this rapidly growing phase, where the brain is very, very plastic, that we're introducing a lot of redundancy into it, that problems at that age necessarily impact on later on in life? Because it is the most capable of repairing itself at that stage.
Yeah.
And it may be that actually problems in that period can be repaired very quickly, and so that is also an added advantage to having this short period of very rapid brain growth for our species evolutionarily.
Yeah. No, that's a good point. I guess the question is, and I just don't know enough about this, and I'm not sure anybody does really, is if you're under nutritional stress, given that you have this very high energy need in the brain, does that constrain, for instance, the proliferation of synapses? Do you have fewer options within your brain to work with? Is that one possibility? I mean, I think what you're talking about might more allude to the second point, that you could kind of lower those costs and delay the process and stretch it out or defer it until a later age. And yeah, so I think the second point is sort of what you're saying, but I think that that might have social ramifications. So if you're developmentally delayed in some way, presumably that's going to have social effects that, you know, might not be good.
Questions and answers short so we can.
Okay.
Alan, can you introduce yourself?
Alan Roses, Duke. One of the things that you said was about evolution. Evolutionary is essentially a genetic matter. Different genes are associated with different changes that occur over time. In 1993, when we found that ApoE4 gave the onset of Alzheimer's disease earlier if you had 2 doses, a little later if you had 1 dose, and later still if you had no doses. A couple of years later, very akin to what you've shown, a psychiatrist named Ryman from Arizona decided to do PET scans. And he decided to do an experiment, which was a remarkable experiment. He took people who had the 4/4 genotype, who were 50 years of age, didn't have Alzheimer's disease, but their estimate was that they would get it earlier than people who had 3/3. And he compared the PET scans of those people. And what he found, and again, this is pre-anything, what he found was there was decreased glucose utilization in areas of the brain that were related to later developing Alzheimer's disease. He's taken that now over the years all the way down to 1 year of age. And those differences are there as a result of a single change in a single amino acid down to 1 year of age. So what you have is a genetic relationship between specific cellular metabolisms, very much fits your long-term deficits and higher cortical functions are developing. When they develop, doesn't have to be just during the periods of growth. It can be growth because the dial has been reset. And that's also seen in lemurs, which are a primate, but not widely looked at. We have a lemur center at Duke. And the largest lemurs, about 65 million years old, are the aye-ayes. And the smallest lemurs are the mouse and the dwarf lemurs, who are incredibly smarter than the aye-ayes at a very small size, but still have a larger brain to mass than in the lemurs. And if you look down their evolutionary tree, changes in the brain occurred in the exact same molecules as involved in ApoE4 and the gene next to it, TOM40. So there are some specific events that could form somewhat of a template for looking at this.
Very interesting. I'm not sure, yeah. I don't know enough about ApoE or the protein that this person found, but, oh, okay, we're going to the next.
Dominic Belli from the NINS. If at 5 years old, if you go over the caloric needs and over the glucose needs when giving, for example, parenteral nutrition or exceeding enteral nutrition, what is, due to your hypothesis, what is the use of this caloric intake? Is it mainly for the somatic growth, or do you increase also the ratio for the brain utilization? What do you think about that?
So if caloric intake goes above requirements at this age, where's it going to go?
If you go over the requirements,
yeah,
what would be happening?
Yeah, I think what we see from the literature is that if you're putting on weight, you're going to put on more fat at that age, right? And that's, I think, part of what's going on, and that's sort of the problem that I set up at the beginning. And
so you have a
presumably the
step
brain
limiting for the brain metabolic
sorry?
rate? You have a step limiting for the metabolic rate of the brain, you think,
You have
at 5 years old?
a set limit, yes. I would assume that the brain and its metabolism is fairly canalized.
Yeah.
You know, that the process of brain development, if anything, is quite buffered, and that the other systems, like how much fat you put down and so on, are more labile and work around that. I don't know that, but that would be my intuition. Yeah,
Thank
and that's
you.
what I think the data also point to.
I think, again, we have to cut short the question. Two more questions. One.
Awesome stuff. Awesome work. One kind of, I'm not sure if it's a wrinkle or whatnot, but so there's a large, sort of growing amount of evidence that suggests that actually slower cortical development through that 5-6 years stage is associated with better cognitive outcomes. Which almost, I'm not sure how that fits in.
Yeah, it would be interesting to know if slower cognitive development implies kind of a lower glucose use, right? Stretched out over a longer period of time. And I'm not sure. But actually, one of the big points that comes out of this for me, you know, we've kind of made this composite picture of what a human might look like in a big cross-sectional, you know, data crunching exercise. What we don't know anything about at this point is how variable this is across different kids. Early maturers, late maturers, high SES, low SES, and so on, right? And I think this gets to that question. We just don't know. There's no individual level data on any of this, really, you know, longitudinally. So.
Although you could possibly get some of that, because from that NIH study, you can find cortical thickness values.
Okay, that's true. Cortical thickness,
but
yeah.
related to synaptic density.
That's a good point.
Also, yeah.
Yep. Hey. So Rajratan, Burke, and Cornell. So thanks for a really elegant analysis. I wonder if you've thought about the implications for what Dr. Gazzaniga mentioned originally about the high risk of sports-related concussion in kids. You have this large growth of the head on a small body, and it seems like you provide an important explanation for why that happens, and then maybe a prescription about why kids shouldn't be involved in soccer and football until they're older.
That's a really interesting point. I actually don't even know what the mechanism of a concussion is. You know, what structures are being impacted by that, and whether that's would be related to the synaptic processes that are going on here. But I think it's a good hypothesis for sure. But I don't think most kids are playing football at age 5. I don't know.
At age 8 there.
Yeah,
in the US.
Very short, quick questions, and then let's.
I mean, your theory sounds very convincing, provided that there's a limitation of food. But why do 5 years old not simply eat a little more? Because then they would not be restricted. And that is a simple evolutionary answer to the problem. Let them eat double as much.
Yeah, and I think the answer to that is in the Kleiber line, right? The Kleiber line, what I showed at the beginning, which relates body mass to metabolic rate. You see that there's a very tight relationship between body mass and metabolic rate across species and across individuals. And why that is, you know, there are different hypotheses. One has to do with, there's a model that's been put out that what is limiting in terms of energy use is the distribution of calories, right? So the circulatory system has a structure that leads to energy expenditure that leads to that 0.75 scaling. Well, I mean, any way you cut it, there's a very tight relationship between body size and metabolic rate across different species, right? So there's some physics, there's some physical reason for that. So I assume there's
you can increase that by, I mean, if you start to exercise, so you eat a lot more. You're not forced to be down there. If you need more energy to run around, you eat more.
Yeah.
If you need more energy to actually make your brain grow, why don't you eat more?
Yeah, and nonetheless, you do find that relationship. So I assume that you can deviate from it, you can expend more, but there must be some cost long term, because in general, evolution is kind of honing in on that value and constraining it. So that's how I think about it.
Okay, well, thank you very much.
Okay, great, thanks.
And now we move to the last presentation by Maureen Black from University of Maryland School of Medicine, Baltimore. and we talk about nutrition from the developing brain.
Thank you very much. I'm absolutely delighted to be here, so I thank the organizers very much for the invitation, and certainly thank the other speakers in the audience for a very stimulating day and a half of presentations and questions. As you'll see, my presentation moves us into some additional areas. It moves us to communities of low and middle income countries, and it moves us into some policy issues. So I'm going to be talking about brain development, the role of nutrients, some research related to nutrition and brain development. We'll talk a bit about poverty, and then we'll talk about interventions, and these are community-oriented interventions to promote nutrition, brain development, and function. We start just a bit about brain architecture. As you have heard, there's a dynamic interaction between genetic influences and environmental conditions. And if we think about two kinds of experiences, there's expectant experiences. These are species-specific experiences that contribute to brain development, and we'll show you some examples in nutrition as well as visual images. And then there are what are called experience-dependent, and these are the kinds of experiences that are unique to the individual, such things as why we speak the language that we do. So when we think about the timing of these experiences, one of the examples that we use in terms of nutrition is the formation of the neural tube that happens at approximately 22 days post-conception and is dependent on folic acid and B12. And if you don't have these, then the neural tube doesn't close or is at risk for not closing, and you can have an anencephalic child or one of spina bifida. So it's an example of how intricate nutrition is to early brain development. Now we think about critical periods, which are very tight, and the neural tube closure is used as one. Or we think of sensitive periods, which have rather looser boundaries. And it looks as though in humans that we think more about sensitive periods than some of the critical periods. This is a portrayal that you saw in the last presentation. And what it does is show us the first 1,000 days that has become very much in the nutrition literature. And as you can see, it begins at conception and extends until age 2. And if you add that together, it's 1,000 days. What this illustrates is the tremendous amount of development that you've just heard that occurs during this period of time. Now, if we overlay nutrition onto this, this is an example of the need for iron. And so you'll see that the period of time from 6 to 12 months is a time of very high iron need. And so this is a time when infants are at risk for iron deficiency. At this point, they are having complementary food that is often very low in iron, and they've used the iron stores that they've gotten from their mother. So it's a very vulnerable time, and you'll see a great deal of research looking at this second 6 months of life because we know it's really very vulnerable. And then after that, the iron need, for example, goes down. Now, what you've just heard, and the timing of the presentations is terrific, that those of us who are interested in child development are happy with the emphasis on the first 1,000 days, and we say it's necessary but far from sufficient. And so this green line takes us to what would be the second 1,000 days and would be up until age 5. And there's often a gap in what happens in terms of opportunities for children during this time. in low and middle income countries, there's often not much of anything going on. In Western countries, children are in some sort of child care. So when we think about early development, we certainly focus on this period of time as well, not only the first 1,000 days. So we're going to talk a bit about the nutrients and how they're related to early brain development. We'll go through this fairly quickly. This is some evidence, much of it coming from animal research, and it shows us how various nutrients are related to early brain development. So this is early neuron proliferation, and you can see they range from protein energy needs through fatty acids to iron, to iodine, to zinc. And then when we look at axon and dendritic growth, again, we have some of the same candidates that begin in early gestation, continue to age 2 years. So this is the first 1,000 days. When we look at synapse formation and pruning, again, we see some of the same nutrients that are necessary during this time. We look at myelination, and this, of course, continues much longer, but again, some of the same nutrients that are necessary during this period of time. This slide summarizes the nutrients that are needed during the late fetal and neonatal brain time. The impact of these nutrients varies by the timing and the dose and the duration. And there's a fair amount of research that looks at this kind of work, both in animals and in children. And much of this is in low and middle income countries because that's where the deficiencies are. Now, how do nutrients relate to children's growth and development beyond the neural tube closure? Well, let's take a look at some of this. So these are children of Mayan descent, and on the left, they're children in Guatemala. These are 9-year-old children, and on the right, they're in the US, so they're from the same heritage. And if we look at the line across them, we can see that the children on the left are stunted, and the children on the right are where you would expect for age 9. So what has happened is that the WHO had a multi-center growth study to look at the growth of children from 6 very diverse countries. So they enrolled children who met the criteria that their mothers agreed no smoking during pregnancy and no smoking after pregnancy, and to follow the WHO guidelines in terms of exclusive breastfeeding. These were primarily middle-class children from these diverse countries, and then they followed them longitudinally to look at their early growth. And so let's look at their linear growth. Well, this is what we see. They're from Brazil, Ghana, India, Norway, Oman, and the US. And we see that during the first 2 years of life, there's no difference in their linear growth. So despite very different countries, very different heritages, that they grew the same way. So we learn that children throughout the world grow similarly when their health needs are met. So therefore, there are something like 150 or 180 or so countries that have adopted the WHO growth standards. These are different from growth references. This is how children should grow under optimal conditions, and this is what we use to look at early growth in children. So if we look now at when growth faltering occurs, and these are data from 54 countries. The blue line at the bottom represents height. So, and this represents the first 1,000 days. And I'll draw your attention to what happens at birth. So we see at birth that there are many children who begin already stunted. So already with a linear growth that is lower than expected. And you'll see that it's really during the first 1,000 days that the growth faltering in terms of their linear growth occurs, and then it basically flattens out. So we see children right from the beginning who are behind. Well, let's look at what wasting and stunting look like. So if we look at the child on the left, this child is not wasted. So that means that their BMI is within normal. They're not stunted. So that means they're not 2 standard deviations below the median in terms of height for age. And they're not underweight. So their weight for age is, again, not 2 standard deviations below the median. The child in the middle is wasted, not stunted, and underweight. And the child on the right is not wasted, the BMI is fine, but is stunted and is underweight. Which child is at biggest risk? What do you think?
Risk for what?
Well, this is risk for subsequent development. So we're interested in cognitive development. This guy. So this is the stunting. So this child is at risk. Well, let me show you some of the data, and more risk than the middle child, because this represents the chronic undernutrition. So what we see, this is allegedly a picture of brain development, impaired brain cells by the child who is stunted. These are some recent data, a meta-analysis by Sudfeld, and they did a meta-analysis of both cross-sectional and longitudinal studies. These are some of the longitudinal studies, and so you can see that taken together, and these are adjusted for some baseline situations. These are developmental risk or cognitive development of children during school age who were in fact stunted in the first 2 years of life. So there have certainly been other studies that have shown the long-term consequences of early stunting. If we look at where the burden of malnutrition is globally, you'll see that it's primarily in Africa and in South Asia. So these 34 countries account for 90% of the global burden of malnutrition. Now, if we look at stunting and underweight, what we see is that the good news is that there's been drastic reductions in stunting, both in the prevalence and in the burden, and also in underweight. So there's been a drastic reduction. So that's excellent news, very good news. Here's the less good news. And that is that 24% of the children in the world under age 5 are stunted. That represents 165 million children, primarily in sub-Saharan Africa and Asia. So the depth of the problem is enormous, absolutely a huge burden. And what do we know about stunted children? Well, there's a series in the Lancet on child development, 2 series actually, and a third one that is in process, will be out in 2016, that looked at the number of children who were in poverty or stunted, and then estimated that 219 million children, or 39% of the world's children, in low and middle income countries were not reaching their developmental potential. So that's an enormous burden that the world is experiencing. If we look a little bit at poverty, and you've heard some of poverty so far, so this is a bit of what we know in terms of child development. We've known for a long time that poverty is associated with low cognitive functioning. And basically, we've thought that it's been related to limited access to resources and some poor hygiene with infections and poor nutrition, and the lack of books or opportunities for early learning and for enrichment. But what we've also seen from our neuroscience colleagues, and has been presented here, is that poverty is associated with smaller white matter and cortical gray matter, and hippocampal and amygdala volume associated with early cognition. These are data, I think, that were shown earlier from a recent study that Kim Noble did. So certainly quite concerning at the lower end. This is parental education, and this is left hippocampal volume. So the Poverty Association is really quite concerned. Now, what else is going on in poverty? Well, we know that people living in poverty have what we call co-occurring and accumulated risks. And this is some of what Stephen Matthews talked about yesterday. Children benefit from mild stress. You all deal with stress. If you couldn't deal with stress, you wouldn't be here. So stress is not necessarily a bad thing at all. But what is bad is toxic stress. So this is very strong stress or unremitting stress. And we think that this can lead to dysregulation in the HPA axis. So this is some of what Stephen talked about yesterday in terms of the stress response. Under normal conditions, when faced with stress, there's a release of cortisol to prepare the body for this response. So there's an increase in cardiovascular tone, and then one returns to a homeostatic balance. But if there's a dysregulation, then there's a weathering that can occur, and the inflammatory system can be dysregulated, and one can then be at risk for disease and for both physical and mental illness. So this is a portrayal of the brain architecture that's related in the case of chronic stress or toxic stress, as it's called. So we think how it undermines child development is that it has these non-reversible changes to children's physiology that then disrupts inflammatory signaling and increases their risk for physical and mental illness. We have, as you've certainly heard from some of the very elegant talks, much more sophisticated ways of trying to understand what happens in children's brains. We can look at the cortisol, and we can then also look at blood oxygenation to try and understand some of the functions, some of the structures, and some of the functions that are happening in children's brains. The final one that's listed, this functional near-infrared spectroscopy, is very interesting because you can take it to the field. So these show that the timing is, believe it's what time it leaves the UK and what time it gets to the Gambia and how long it takes to be set up. So, there are now some very sophisticated assessments that can be done looking at brain functioning. It can be also used with infants. So the field really has much that we'll be able to learn from some of the new mechanisms that are available. I wanted to talk about, some of these things are really very hard to study, and we have a history of taking advantage of naturally occurring events. So the Bucharest Early Intervention Project is a naturally occurring event, and what happened is there were 136 children who were abandoned at birth and placed in orphanages in Romania in quite dismal circumstances. There were a number of investigators who had a project that was funded that would randomize where these children were placed. You can debate the ethics of this, but what happens is that half of the children at approximately age 21 months went to foster care, and the other half remained in the institution. At the same time, they had a comparison group of non-institutionalized children, and then they have been followed over time. There are a number of adoption studies, but adoption studies are far from random. Who gets adopted? Who gets placed in foster care? The cute kids, those who were able to reach out and pull from their environment. So they're really, really quite biased. But in this one, they were placed randomly. So there have been 30 or 40 at least studies looking at what has happened to these children, and we learned from this. So these are So I'll show you a few of them. If we look at growth, then what happens, and this is not surprising, that the children who go to foster care grow better within about a year of their placement in terms of their height and weight. Their head size remains low, and their growth is associated with their age at placement and their caregiver sensitivity. So this is not surprising. When we look at their IQs, their IQs of the children who are in foster care are better than those in the institution, not as high as those who've not been. The comparison group that was not institutionalized, but they're also influenced by their birth weight and an increase in height. So it shows some nutritional relations with it early in life as well as subsequently. If we look at their neural development, this varies by their institutional history, and the way that you read this is the group that's labeled NIG, that stands for never institutionalized, so that's the comparison group. And this FCG means they went to foster care, and then this CAU, they stayed institutionalized. So if we look at this, we see that in terms of gray matter, the children who are in foster care look no different than the children who remain institutionalized. So we see that this suggests that the institutionalization disrupted the brain development. They had less gray matter, and there was no catch-up in foster care. If we look at white matter, we see a different picture at this point. So there's no difference here between the foster care and the not institutionalized. So it looks as though then there was a disruption and the foster care children had a catch-up. These are observational, they had an intervention, and in some ways these are hypothesis-generating kinds of findings. This is one of cortisol production at age 12. And on this side, again, you have this is the not institutionalized group here, and these are the two foster care groups. So here you see no difference between the foster care and the comparison in their stress response. So it looks as though the foster care may have attenuated the dysregulated HPA. So that's pretty exciting. So that shows that they have benefited from that. On the right are the two groups in foster care, and they differ by age. So you see an age difference there, and it looks as though the intervention effects occurred for those placed prior to 24 months, suggesting a sensitive period, again, within that first 1,000 days. So these kinds of, this information from the Bucharest study can be really quite fascinating, and it suggests that what development looks like in a general sense is that if you're raised with what we call protective factors, then really at birth, and this represents birth, this represents a prenatal time, again, of the first 1,000 days, that you're better off and your development continues. If you're born with these risk factors, then the disparity occurs at birth, and basically it continues and actually widens. The blue represents an intervention, and so what the data suggests is that interventions can be beneficial, can in some ways pull one closer to the green, in some ways the disparities remain. So the question is, when does it happen, and really what do you do in terms of the intervention to try and reduce the disparities that occur and reduce the 39% of children who don't reach their developmental potential. So we think about nutritional interventions, and if we intervene with moms, then we can intervene in terms of improving maternal nutritional status. We could do that prenatally, and what we've learned is that prenatal may be too late, and so we can do that preconception. And there are those studies that are in the field now looking at those timings. With children, we can intervene by promoting breastfeeding, and there's evidence that breastfeeding is, particularly exclusive breastfeeding, is beneficial to children in terms of their growth and in terms of their development. We can look at complementary feeding, and that's, those are the kinds of foods that children we used to call weaning foods. We can look at single micronutrients, we can look at multiple micronutrients, we can look at proteins and calories. And there are many ways of doing this. You can do this with supplements. You can do this with fortification. So that's adding these, some of these micronutrients to food, and that's often how we do it in the West. This is a conceptual framework that sometimes we use thinking about early child development. So we've been talking about brain development and function and how that maps onto children's development. And there are biological factors. This is the nutrition that influence brain development. And there are poverty and sociocultural factors that influence brain development. We know there are also caregiving factors, and certainly the Bucharest studies have shown us that, as well as a number of other studies. And we know that those include things like responsive caregiving and early learning opportunities. And what happens in many of our studies, what we've done is we've just adjusted for these poverty and sociocultural factors and caregiving factors. They're kind of pesky, so we get them out of the way. But what happens is that if we adjust for those, then we have given up looking for additive or synergistic effects of nutrition and caregiving. So if we don't consider the context, then we may actually be missing what the nutritional interventions do. So what we suggest is that rather than getting rid of those, that maybe we should study the caregiving factors and the nutritional factors, because we know that children's development is influenced not only by nutrition, but we know it's influenced by the environment, by the caregiving environment that they're in. That's represented somewhat by poverty, but it's represented by the early learning and responsive caregiving. So those are some of the studies that certainly have been recommended. This is a portrayal of how you might think about some of the responsive caregiving. There are prenatal threats and postnatal threats, and then this buffering effect of responsive caregiving. And we think that that may influence the immune system and then influence brain development. There's a study in the field now that's called malnutrition and enteric disease. And what they're looking at is how enteropathogen infections relate to intestinal inflammation and how that alters really gut function and whether that's related to undernutrition and cognitive development. This is a study that is taking place in 8 low-income countries. It's funded by Gates and spearheaded really by the Fogarty at NIH. So the data from this study are just starting to be available, but certainly interesting looking at the combination of inflammation and some of the things that we've talked about. I'm going to show you very quickly a study that we've been doing in India, and very consistent with the last talk, we're focusing on children in that 3-year age range, not looking at, this has an analog where we looked at children in the first 1,000 days, but these are the preschool children. We do this study in India because that's where the problems are. India has a system of the integrated child development services where they have preschools or Anganwadi centers throughout the country. When you have a population of 1.2 billion people, you have 40 million children. So we worked with, the study takes place here in a new state called Telangana. It used to be Andhra Pradesh. And we collaborate with the National Institute of Nutrition. And we work with these government agencies because if things work, then you have a possibility of actually scaling up a project. So I'll show you very quickly how it worked. So our research design was child care centers or preschools come in all different qualities. So we characterized low-quality preschools and high-quality preschools, and then randomized that they got a fortification of either micronutrients or placebo within each. These are the baseline characteristics of our sample. And you can see this is the issues that over a third of the mothers are anemic. And when we look at the 3-year-old children, 20% are wasted, 40% are stunted, that almost a half are anemic, high rates of iron deficiency. So the problem is absolutely there. It's a very straightforward design that we have a baseline and an end line. It's an 8-month study, and the preschoolers received a midday meal fortified either with micronutrients or placebo. This was the fortification, which was developed by the National Institute of Nutrition in India. So they have iron and zinc and folic acid and vitamin A and C and B12 and B2. It came in these packages given with a spoon, and the fortification was mixed in a small amount of food, in 100 grams of the midday food. So this is what it looked like. The food that the children ate was a rice and dal, lentil, veggie mix. So this is the food. These are the children having the food. The 100 grams is a very small amount. That's what had the micronutrients in. Once they ate that amount, then they could have the rest of the food. But that way we ensured that they actually got the food. So when we look at what happened in anemia prevalence, then we see a dramatic decline. Their rates of anemia declined from almost 50% down to under 10%. If we look at the change in the biomarkers of iron deficiencies, again, you can see dramatic changes in ferritin, in transferrin receptor, and in the transferrin receptor log ferritin ratio. So certainly they got the micronutrients and had a dramatic change on their micronutrient status. When we looked at their development, we looked at some markers of early child development. So we looked at language, at visual perception, at motor. Inhibitory control is a measure of executive functioning that we looked at in these 3-year-olds, and then we had a rating of the caregiver rating the child's behavior. So in the beginning, we saw some differences between the low-quality and high-quality preschools. No differences by those randomized to get the micronutrients or not. When we looked at the expressive language, what we see is an interaction between the quality of the preschool and whether they got the micronutrient or the placebo. And basically what we find in the high quality preschool, there were no differences on their language. But in the low quality preschool, you see an increase in their expressive language among those who got the micronutrients and a decrease among those who got the placebo. So there was a benefit in the low quality, but not the high quality. And actually the benefit in the low quality then reduced the disparities in the children. They look then no different from the high quality kids. And this is adjusting for baseline and so on. When we looked at inhibitory control, you see a similar pattern in that you see an interaction between the two, not main effects. And so when we look at it, again, you see the difference in the low quality, that those who received the micronutrient had much greater benefit than those who received the placebo. So again, the micronutrients improved inhibitory control in the low quality preschools. And the similar pattern in the caregiver report of the child's behavior, those in the low quality showed a benefit, those in the high quality didn't. So a summary was that when you consider context, you have a richer story than if you don't consider context. So there's an emphasis on integrating nutrition and early parenting interventions. For children who are under age 3, this usually occurs during home visiting programs. And the evidence suggests that impacts are larger when parents and children are involved together and when it's an active intervention, and that targets the most disadvantaged children. Again, like our low-income preschools, that's where you see more of an effect. The expressive language, when we look at early language environment, we see really a paucity of language. So I'll show you the way that we stimulate language, and that is by making books. And when we make books, they have no words. So they have no words because often mothers are illiterate. So when you have them, they include both descriptives of what you see, but they also include affect. And then mothers are trained to talk to their children about the books. So it's a storytelling, and the books provide a stimulus for the storytelling. These are some that we've used in Guatemala. These are some that we've used in India and Bangladesh. So they're easy to make, but they're very consistent with the culture. In the 3 to 5 year olds, again, it's integrating nutrition and preschool. When we think about the cost of the intervention that we did in India, the cost of the micronutrients, it is minuscule because it's adding it to the food, and really the cost is the distribution to the preschools. So it's an extremely low-cost intervention for an incredible benefit. This is a meta-analysis looking at a number of nutritional interventions. It's a bit hard to understand because there's so many of them that are here, but you can see that there's an overall benefit looking at measures of mental development. This is looking at psychosocial development, and again, you can see a benefit. So when you put them together, it's an even stronger one. This represents a life course that we need to have interventions not only early, but that continue through early childhood and then on through adolescence to ensure adult health and well-being. 2 studies in Science last year that were long-term outcomes. The one on the left is from North Carolina. It's a 35-year outcome of an early childhood intervention, and what they found were benefits on blood pressure and metabolic syndrome. They also found benefits on college attendance. These were very low-income children in North Carolina. We also had one in Science from an intervention in Jamaica after 25 years. And they found beneficial effects on wages, as well as less likelihood of being involved in violence. So strong effects from very early interventions. What has happened in terms of early child development is that there's absolutely been a revolution. There's been tremendous attention to the Millennium Development Goals, or what we now have as the Sustainable Development Goals, and as of last month, they've been renamed to the Global Goals. Well, these are statements from the Secretary General of the United Nations, emphasizing early child development as a public priority. This was never thought of before. Early child development was nowhere on the radar. So the attention that has gone into investing in young children and investing in early child development is monumental. 40% of low and middle income countries have policies related to early child development. So this is really a time the brain science has certainly contributed to this revolution, and there's really much to be done. So this is our team in India, and thank you to some of the donors. This is an issue of the Annals of the New York Academy of Sciences that contains 20 papers on integrating nutrition and early child development, and I thank you.
Yes, Maureen, thank you very much. Yet again, a very high emotional content in the presentation. I will leave the floor to more expert questions, but can you give us briefly the ethical framework of those controlled and placebo studies, and how do you see this evolving, and whether you see perhaps even better options to avoid, of course, what might seem a great dilemma?
The ethical control in these studies is certainly one that is a serious one, and going through ethical control in India is far more difficult than going through it in the US. And the US will not give permission until India gives permission. It took us 1.5 years to get the permission to do it. You know, I think what happens is there are certainly studies that will show giving iron does not have an effect. And so it's not absolutely clear that how these studies work. And as you could see, in the high-income children, there was not an effect. I think part of my own sort of way of dealing with it is dealing very much with our Indian colleagues. They are true partners in this process. So it's not parachuting in at all. And we go through the ethical standards just as we do with any study.
I'm a little bit surprised that you say that early childhood has not been on the radar screen. When I was in training in medical school, which was like a century ago, the spit studies.
Yes, the spit studies.
The Harry Harlow modeling of that in monkeys. The later Romanian orphanage studies showed how absolutely essential, even if diet is controlled for, interpersonal interaction is, and how devastating the consequences are.
Absolutely, you're absolutely right. Those studies are really landmark studies. But what's happened in terms of investment in early child development, the investment has really not been there. It's not there in the U.S., and it's not there. There's been very little public policy reaction, very, very little investment. So that's what's coming. But you're absolutely right. The evidence has been there for a long time. Yes.
I admire what you said, but in this period, which you have described, another major public health intervention occurs worldwide, which is quite well regulated. I worked a lot on it, and that is the vaccination.
Yes.
Because we are so afraid of global epidemics. I would like you to comment a little bit on it, since that is an obligatory and legislated intervention in all of the countries WHO deals with. And when I developed the triple vaccine, which, you know, reduced the largest killer of these children in the first 1,000 days, whooping-cough was the largest killer, and we reduced it by 70%. There you have an intervention where we measured anemia and all this. Would you comment on how the two could be coupled? Because that is legislated. This depends on caregivers. Bill Gates and Melinda Gates may fall out and they decide not to fund it, and so on. So vaccination occurs exactly at this time. Infection rate data is well known for this time. Would you comment on how they do interact, please?
Thank you. It's a wonderful comment, and I absolutely agree. You know, there was a statement spearheaded by Gavi in the Lancet Global Health last year that I was part of, and others who were part of, who were interested in child development, were certainly part of. What has happened is that the head of the UN, who has talked about child development, along with the heads of WHO and UNICEF, and where the combination is to putting what the health sector does, including vaccines, having child development be part of that as well. And so your comment is really right on that. You know, when we talk about the implementation in low and middle income countries, often things are very sectorial. So there's a health sector, and the nutrition sector has certainly increased, but the child development sector is sort of nowhere. Is it part of education? Well, not really, because the educators tend to begin at primary school, and their indicators are all about primary school. So early child development suffers by not having an indicator. The closest indicator is stunting, and child development people are not satisfied with stunting. But so your comment is really right on, and there's a... Yes?
Which are legislated and which are international.
I agree, but what happens is child development is more than a vaccination or more than a single shot. So how do you hook it? And there's a new Lancet series that is coming in 2016 that is very much how to put child development and health together. Right. So thank you.
Suzanne Suter from Nestle Foundation. Thank you very much for this broad overview. The projects we sponsor, we support, the majority of them are exactly like the one you mentioned. And I would not, of course, challenge the fact that stunting imposes a big health risk on the children. But still, with time, when looking at the WHO growth curves and definitions, if you go back to the number of countries taken initially in order to establish these growth curves, I think the bunch of countries was rather small.
Mm-hmm.
And I always had problems accepting these growth curves for every population around the world. And I think in a period where malnutrition and obesity are major problems, both, you need to be correct with regard to growth curves. So right now there is a group between workers in Sri Lanka and Harvard which challenges the validity of the WHO growth curve, the international growth curve. And I think it's very important that we can rely on standards everybody can have faith on.
Mm-hmm. No, I certainly agree with your comment. They were a step forward from relying on either local references or the references used in the U.S., but it is true. It is striking in these very varied countries that you saw similar growth, but as with many things in science, it's a step forward, not necessarily the final step.
Last question there.
Noel Solomon, Guatemala. There were so many lovely elements. I wish I had time to comment on many of them. With regard to Stefan's question on ethics, you failed to mention that there are some studies in which the iron treatment was actually detrimental.
Yes, that's true.
We have to have a placebo because we never know when we're doing harm.
That's true.
The second point about stunting, everybody who knew anything raised their hand that indeed stunting had the worst outcomes. But I'd like to emphasize, at least in my opinion, that it's not that they were small. As a matter of fact, stunted children do not have smaller heads, and we should make that point. It was what making them small, the stresses and the adverse circumstance that made them small, which gives them the adverse long-term outcome. So size matters, but in this case, it's what made you small, not being small. That's the outcome.
Well, what I would say is that there's also an outcome of being small. And what happens, what are the consequences of being small? The expectations on a child who is small are reduced. So the expectations from a teacher or from a parent or from a peer may be reduced to the child who is small. The child then perceives of themselves as being potentially less competent, less able to do things. So there's not only one pathway that leads leads to...
And finally, the EED, I'm glad you brought up the enteral, because it is a tremendous inflammatory and infectious stress. And this links back to the previous paper with regard to that period of life and the consumption of glucose. We forget that inflammatory cells are also among the glucose-dependent in the body. So if we have a brain and an inflammatome that are equal size, there'll be competition for the glucose. Thank you.
Thanks.
Very short. Jim Kaput, Nestle Institute. So we're concerned about the fact that some kids don't get the vitamins because they're in the control group. What we're looking at is inter-individual variability. So you put everybody in the intervention group, and then you ask how individuals respond. You still can do population level data from baseline to post-intervention, but then you take the same data and you say, how do they start and how do they respond? So you can do both at the same time without losing any scientific credibility.
Certainly, there are other designs to doing that, and it is a question. One of the things that we do also that I didn't mention is that all of the children receive the will receive the fortification for the next year. Again, the fortification is very low income, so it's just the distribution cost. So, yes, that's a good point.
Well, thank you very much.
Thank you.
I'm sorry, I just would like to add one word for Solomon's comment, and just to clarify my position. I wasn't necessarily, at least at this stage, challenging the ethics. I just was asking what is the framework and how you see this evolving. And what Jim has done, Jim's comment goes exactly in the direction of what I think, that we should design more innovative, more personalized ways of addressing the same questions without necessarily making those splits. All right. Thank you, indeed.